Hello, my name is Dr. Veronica Hood, and I'm the Scientific Director at the Dravet Syndrome Foundation. Thank you for joining us today for the second session in our three-part educational webinar series on AAV-mediated gene therapy for developmental and epileptic encephalopathies. This educational series is provided by the Dravet Syndrome Foundation in collaboration with Encoded Therapeutics and is intended for healthcare providers. This educational series will be led by two experts in the field, Dr. Sarah Weckhausen from the University of Antwerp and Dr. Michael Lawlor from the Medical College of Wisconsin. If you have not yet had a chance, please watch the first session in this series in which Dr. Lawlor introduces the basic concepts of gene therapy and AAV-mediated gene therapy. A recording can be found on the Dravet Syndrome Foundation's website. Please submit any questions you may have through the intake form on the DSF website by December 15th, and we will post responses in March. During today's session, Dr. Lawlor will discuss both translational and clinical considerations of AAV-mediated gene therapy. Dr. Lawlor is a professor of pathology and associate director of the Neuroscience Research Center at the Medical College of Wisconsin. Dr. Lawlor directs two clinical laboratories that process and diagnose muscle and nerve biopsies, and his research group has many industry collaborations on congenital myopathy, muscular dystrophy, mitochondrial disease, and gene therapy. And with that, I will turn the session over to Dr. Lawlor.
Okay, so for part two of this series, I'd like to talk a little bit more about translational and clinical considerations of AAV-mediated gene therapy. And so once again, this is based in my experience as a muscle pathologist in working with a number of pharmaceutical companies that are performing AAV gene therapy work. And in general, this talk is going to focus on some clinical efficacy and safety findings related to current AAV gene therapy studies, and discuss some of the positives and negatives about the current state of the approach. And so just to start off, I think as a good continuation of the last talk, so AAV gene therapy is becoming a clinical reality. There's a number of active trials for a pretty wide variety of disorders that are currently in the clinical trials stage, and there are two FDA-approved gene therapies. So one is Luxturna for a subtype of retinal dystrophy, and then Zolgensma is a gene therapy related to spinal muscular atrophy type 1. In addition to those two approved products, there's dozens of AAV-mediated treatments in clinical trials, and over 3,000 patients have been treated with gene therapy in the last 21 years. And so we are definitely getting a good look at what this sort of technology looks like in humans. As we'll talk about later, we're also getting a good idea of the fact that some questions related to immune responses and safety can unfortunately only be answered in humans, as animal models don't seem to be predictive of some sorts of responses that we're seeing in patients. Now, in designing a gene therapy study, there's some elements of it that are very similar to designing a traditional clinical trial study, and others that are somewhat unique to gene therapy. And so just looking at the potential stages that one goes through. So first of all, at the preclinical stage, this is often focused on in vitro and animal work in association with identifying whether or not a gene therapy product is working appropriately, informing on safety and efficacy, deciding on doses, and also endpoints related to how you determine safety and efficacy, and then to assess for potential immunological responses. Now, as we'll talk about in a minute, these animal models are not necessarily ideal for predicting all of these endpoints and responses, and so this needs to be taken into context and reanalyzed in light of the human disease and the clinical trial experience. Another stage, which often can be performed in parallel, is human studies of patients with the disease. These are called natural history studies, or clinical studies of series of patients. And the idea here is to understand a knowledge of the disease state in humans, the progression in humans, whether or not there's any differences between the humans and the animal models, whether or not the approaches used in the study of animal models are even applicable to the type of setting you would need to study human patients or human tissue. And so the idea here is to really translate things over and establish expectations for the clinical trial based on the combination of the preclinical data and these human studies. And then really you get to the clinical trial stage. And so you want to look at, obviously, outcome measures. So you get, so if it's a motor disease, you want motor outcome measures. If it's a cognitive disease, you want cognitive outcome measures, things like that. In some cases, the variability of the human disease is pretty large. And so you may end up really being restricted to comparisons of the patient before and after therapy within a certain specific subset of behaviors. In some cases, there are molecular tests or tests of tissue that'll tell you more reliably whether or not anything has changed in relation to the disease than some sort of behavioral or cognitive test, because an individual patient's performance on it could be highly variable. And so all these need to be taken into account when you design the trial. And in addition, you need to establish ways of determining how much of the gene therapy product is being distributed and expressed within your target tissues. And so, all of these come together to evaluate safety and efficacy. Ideally, the efficacy endpoints relate to the disease phenotypes. Safety, obviously, is something that gets declared also in patient phenotypes and in laboratory values. And then things like biodistribution, transduction, some of the biology can be performed in tissue studies or in studies of body fluids and things like that, depending on the disease. Now, one other consideration of how the immune response in gene therapy can affect trial design that's a little bit unique to gene therapy, or at least gene therapy compared to traditional therapeutics, and that's the potential for immune responses. So as I covered in the last lecture, there's the potential for the virus to produce an immune response. There's also the potential for this new protein that's being produced by the gene therapy to produce an immune response. And while the preclinical models are useful for predicting this, so if you saw a significant immune response in a preclinical model, you would definitely be worried about seeing it in humans. At the same time, a lot of preclinical studies showed no significant immunological response. And then when some of these studies are progressed to the human clinical trial stage, it turns out that the human patients have an immunological response that was never observed in the animals. And so we need to kind of have an understanding of the need to do some of this learning based on at the human clinical trial stage, rather than just checking for efficacy when we're talking about gene therapy. The other thing to keep in mind is that, as we'll talk about, there is a limitation in efficacy that can be produced if you've got antibodies against the virus being used for gene therapy. And so just the potential for this prevents, first of all, the dosing of healthy patients that may need gene therapy for something else later in life, or even a conventional dose escalation strategy where you'd start at a dose that was definitely too low, and then you would gradually go up to something that reached efficacy. Because anybody that gets a high-dose gene therapy, for instance, is going to develop an antibody response to those AAVs, and then you won't be able to treat that patient again. And so really these studies need to be designed in a way that even the lowest doses used should have some sort of expectation of efficacy. So in the context of our discussion today, I'm going to talk about really three main trials. The spinal muscular atrophy trial has been previously published, and we've got permission to talk about the X-linked myotubular myopathy trial and the Sanfilippo syndrome trial. In the interest of full disclosure, I should say that I'm extensively involved in this X-linked myotubular myopathy trial. But what I will be saying about it is all publicly available information. So at first we'll talk about efficacy, and then we'll talk a bit about safety as well for a more balanced discussion. And so the idea here is that these are three really good examples of the potential efficacy that can be observed in a gene therapy trial, as well as the potential for safety concerns. And so, as I mentioned earlier, Zolgensma for SMA type 1 is an FDA-approved product. The AT132 for X-linked myotubular myopathy is still in trials, and we'll talk a bit about that later. But both of these are severe neuromuscular disorders that cause significant weakness and death in very young children. And so both of these trials were actually centered around the idea of the ability to achieve motor endpoints that untreated children were not able to achieve. And so in comparing the efficacy or demonstrating the efficacy across both of these trials, you can see this CHOP-INTEND scale, which is a functional scale performed on children. Normal children can usually max out this scale by about 9 months of life, but you can see that the untreated children really do not have particularly good performance on the scale, and they hit the middle to the low end of the scale. And that patients that are treated with either Zolgensma for SMA or AT132 for X-linked myotubular myopathy have a very different trajectory of their acquisition of motor function in comparison to the untreated patients. And essentially in both trials they maxed out the CHOP-INTEND scale, and then further gains in function needed to be assessed in a different way. Similarly, also, both in SMA and X-linked myotubular myopathy, the efficacy was also demonstrated by the fact that these patients post-treatment were able to achieve motor milestones that weren't really observed in the untreated patient cohort at any point. And so these are examples of really severe diseases where patients don't have the ability to reach normal developmental milestones, and so the acquisition of these things is a very positive sign that the disease is being reversed or addressed in a way that's actually a bit harder to do for disorders in which patients start out with normal function and then decline, because such declines are a little bit harder to predict. And then additionally, in the myotubular myopathy trial, there were also very large decreases in ventilator dependence, and Zolgensma had excellent respiratory recovery as well, from what I've heard. So really, in terms of the potential efficacy of AAV gene therapy, when it is safe, when it does not elicit a negative response, it can have an extraordinary effect on a patient's development and life. Similarly, when you look at Sanfilippo syndrome, this is a disorder in which the natural history has an element of cognitive decline. And so when you look at the dosed cohort, you have patients not experiencing the cognitive decline and staying within the normal developmental range for age. And so again, you have a disease trajectory that's very concerning and very severe, and that the gene replacement therapy is capable of giving that patient a different trajectory that's much more similar to what the non-disease state would look like. So in relation to safety and looking at the same three trials, with respect to Zolgensma, we have over 1,000 children that have been dosed on this trial. The overall safety signal on this is favorable, and while there were 2 deaths observed in association with Zolgensma dosing, there were neither of these were thought to be associated with treatment. Safety profile on Sanfilippo syndrome is also very favorable. 19 patients have been dosed. There are no deaths or treatment-related SAEs in that, and so that's also very encouraging. The AT132 study, however, highlights some of the dangers of gene therapy and the working in the severe disease arena. And that's because there were a subset of SAEs, but most concerning is that 4 patients at this point have experienced progressive cholestatic disease that progressed to liver function and patient death in this study. Based on subsequent investigations, this was judged to be a worsening of a pre-existing liver phenotype that had been not completely recognized in X-linked myotubular myopathy, and that the gene therapy really pushed it over the edge in terms of being a progressive liver failure phenotype, whereas before it was, in most patients, a very minor phenotype that wasn't well understood or studied. And so, from one perspective, this is a disease-specific safety issue, but it also highlights the need to understand as completely as possible what goes on in the human disease, and to be cautious about expectations in the study. So while this study represents a high point in terms of efficacy, it also is concerning in terms of safety, and it is difficult to look into some of these phenotypes when the animal models don't necessarily share all of the disease phenotypes. And so at the moment, Aspro is currently on hold while this investigation continues. One final note here is in relation to the duration of treatment. So the duration of all 3 of these treatments has been excellent so far in patients in whom it has been effective, with Zolgensma showing 6.2 years and then lower numbers observed for these other agents. What I can say here is that this is how long these have been around, and so this isn't necessarily the total duration of treatment. This is the duration that treated patients have existed, and to my knowledge, there aren't any patients in any of these studies in whom the efficacy of treatment has diminished over time. So the maximum duration of treatment in relation to these 3 programs at least has yet to be established. So in transitioning to the current challenges associated with gene therapy, one thing that I mentioned earlier was the immune system. And so immune responses are difficult to predict, and they can be broken into a couple of different separate concerns. So one large thing in relation to worrying about how patients will respond to dosing, or whether or not the dose will be effective, is pre-existing immunity. So for instance, neutralizing antibodies that will bind to AAV, then you dose the patient with a bunch of AAV, but if there's a whole bunch of antibodies in the bloodstream, then those antibodies will bind to the AAV and they won't actually hit their target tissues. And that's obviously a big concern. Now, in the context of when you provide the gene transfer, there's the possibility of a fairly immediate activation of complement in response to the AAV infusion. And so that has the potential to cause a subset of immunological reactions that can lead to specific types of problems with blood vessels and kidney damage, things like that. Further on, you have this potential for the traditional immune response to both the virus and the transgene product. And so you have the potential for cytotoxic T cell responses. You have the potential to develop neutralizing antibodies to the AAV, antibody responses or immune responses to the immune or to the transgene itself. And so all of these things need to be taken into account. And of course, it's often an open question about what to do about any of these either long-term pre-dosing or long-term post-dosing events, as this is going to affect who you include in your study and your adaptive immunity response, and how you can modulate it is going to affect any kind of approach towards the redosing of patients. So to drill down a little bit more into neutralizing antibodies, so these are the antibodies that patients might have before dosing that would bind to the AAV. So there's been studies done of the prevalence of anti-AAV antibodies, and really, as you can see here, there's a big age divide. And so if you're dealing with a young patient that's under 8 years old, there's a relatively small number of patients that will be positive for anti-AAV antibodies. And yet, beyond the 8-year-old point, there is a very large proportion of patients that will be positive for anti-AAV antibodies. And so this definitely clarifies the necessity of developing strategies to deal with these kinds of pre-existing antibody patients, because you don't want to lose the ability to enroll half of your patients on the basis of neutralizing antibody status even before you start your treatment. That's a lot of patients to leave out of a study. It's a lot of patients to leave untreated. And obviously, 8 years old, that essentially excludes all adult diseases as contenders, as well as a large number of pediatric diseases. And so there's a number of experimental strategies that are in progress to help understand how to overcome these challenges. As mentioned earlier, it's a little bit difficult to predict the best approach, because in many cases these immune responses that people are most worried about in humans aren't actually being modeled correctly or predictively in animal models, just on the basis of species differences. In terms of how you use neutralizing antibody amounts, there's really very little consensus on how to do it. And so what you can see here, this is just a collection of trials that are currently ongoing. And you can see here that a number of them have an neutralizing antibody cutoff, so a titer at which they say, okay, this amount of neutralizing antibodies or above is too much for us to consider dosing the patient in the clinical trial. And then there are other studies for which there isn't a set limit for it. One thing to note is that a lot of the studies for which there is less concern about anti-AAV antibodies are in the central nervous system, because the studies that are really focused and dosing in the central nervous system are thought to be less limited by circulating antibody amounts, as the agent is assumed to be able to get through the central nervous system and then eventually will come out into the periphery, but that there won't necessarily be a massive limitation of efficacy in antibody-positive patients. Overall, the consensus here is that we're still trying to figure out what to do with these neutralizing antibodies and what thresholds are safe, which ones aren't, and of course, how to take patients with a less desirable neutralizing antibody profile and then still treat them effectively with gene therapy. So another issue to discuss is immune toxicity and inflammatory toxicity. This has been seen in a couple of studies, but as I mentioned earlier, is a bit difficult to model outside of human patients. In general, their AAV vector immunogenicity is low, but the impact that this immunogenicity has in part depends on the target organ and the disease that you're dealing with. And so an additional risk factor can be the dose and total exposure that you're using. And so in cases where you are dosing on the basis of weight and then you have an old or a heavy patient, you're giving very, very, very large doses, and that can increase the potential for the immune system to respond to the dose itself. And while some of these things are related to the disease, there are others that can be mitigated by vector design, capsid design, things like that. And unfortunately, as mentioned earlier, the impact of some of these things is hard to assess until you really try them out in human patients. So overall, I think key things are to be cognizant of the design of the vector and also the route of administration. Because as mentioned earlier, if you're trying to get, for instance, into the central nervous system, some of these things can be avoided as potential toxicities because doses can be kept low if your route of administration allows you to really specifically get the AAV into the central nervous system. And then the other thing to mention is that as we've gained more experience in observing and recognizing immune-related reactions to AAVs, there are certain things that are class effects that are seen across multiple programs and not necessarily related to the transgene. They tend to occur within a limited time scale after dosing. And at this point, there are increasingly useful treatment, immunosuppressive, steroid type paradigms that people can use to mitigate the impact of a response when one is observed. Now, one example of this sort of response is can be seen in one of the hemophilia studies here, which is shown. And so there is the potential for T-cell responses to occur and to cause hepatocyte damage really between 4 and 8 weeks, as you're seeing here with this increase in liver function tests. And in the subset of some studies, for instance, this factor IX study, this was also related to a decrease in the efficacy of the agent. So you can see the factor IX expression was doing quite well. Then you get liver damage related to the increases in the AST and the ALT, and then over time the efficacy of the gene therapy as measured by the percent factor IX activity goes down, presumably related to the destruction of the liver cells. And so, a few things to mention. So as I mentioned earlier, this is an example of one of these class effects where the elevation of LFTs several weeks after gene therapy is a relatively common response in AAVs that can be mitigated with steroid treatment. In a study like this, where the target tissue is also the liver and the response may be destroying liver cells that have received therapy, this can obviously affect the long-term duration and efficacy of the product. In a case of, for instance, a muscle gene therapy, it's less catastrophic for efficacy because while you have this increase in liver enzymes, there doesn't necessarily need to be any sort of destruction of muscle tissue related to that, and so you may not lose efficacy at the same time as the liver is undergoing this sort of response, and especially as it's managed successfully. One further caveat is that this is the type of study in hemophilia that you're able to look at a serum marker and evaluate efficacy during the progression of these responses. In many of the gene therapies that are being evaluated right now, that's not actually possible. So for instance, in the muscle studies, you get muscle biopsies. Muscle biopsies aren't necessarily taken at the same, nearly as early as what you're seeing here in the 4 to 8 week time period. And so the impact of some of these enzyme spikes on efficacy is something that is less definitive in those studies in comparison to these hemophilia studies. Other potential organ toxicities can be observed. We already talked about the Astellas gene therapy trial and the progressive and fatal cholestatic liver dysfunction as a component of a progression of likely pre-existing hepatobiliary disease, or at least a hepatobiliary disease tendency. There are other studies in which liver injury has been observed. There are several microdystrophin studies by companies that have reported thrombotic microangiopathy, which is a complement-related response, which, if you'll remember, is one of those responses that happens very quickly after dosing. And then other adverse events on which less is known about the etiology and that require further study. Now, as I had mentioned earlier, one of the considerations when you're trying to balance safety and efficacy in gene therapy is the route of administration. And so when you think about the target tissue and the route of administration, there are some situations when you're looking at, for instance, the eye or the brain, where you can get by with using a very small dose and really essentially avoiding immune complications if you can figure out a way to distribute it to those organs before it goes into the systemic circulation. In comparison, when you look at liver and muscle, particularly muscle, you have a large tissue amount, and you have the requirement to distribute it very broadly through the body, and so the dose that you're dealing with is going to be very high in comparison. And so these factors need to be taken into account when you design the trial, obviously, but also in terms of risk mitigation and the types of things that you have in place. As I mentioned in my last talk, there are ways to improve potency and where the transgene is being expressed, and so these are all things that are active areas of development that will take, I think, the current generation of gene therapy efficacy and safety and move them dramatically forward as we start integrating some of the things that are in development right now into some of the programs that are moving forward at gene therapy companies. In relation to the brain specifically, there are a variety of investigational gene therapies that are being developed for CNS diseases, and there's also a variety of potential approaches for the specific dosing of the central nervous system versus the systemic circulation. And so when you look at a traditional intravenous administration, you really don't get much distribution to brain. You get a ton of distribution to the liver and to many other organs. And even if you have a promoter that doesn't express outside of the brain, the doses that you would need to get effective brain biodistribution have the potential to be toxic in these other organs. And so obviously, for something that's like a brain-specific disease, an IV route has high dose requirements, limited brain distribution, and then of course AAV antibody status. If the patient has neutralizing antibodies, then it would be even harder to get efficacy in that. In comparison, all of the central nervous specific routes have a couple of real advantages here. So all of them are comparatively low in dose, and all of them are good at avoiding a significant impact of neutralizing antibodies. And so that is a big big improvement when you know that your target tissue is in the brain. As you can see here, there's some level of preference to other routes of administration versus intrathecal administration, but specifically what is most appropriate for a situation is likely most dependent on where the disease itself is manifesting itself and how best to distribute to the specific cells in the central nervous system that are most affected by disease. One other thing to mention is that you don't entirely avoid the periphery in terms of biodistribution when you provide dosing to the brain. So you end up dosing the brain, goes into the CSF or in the brain parenchyma, and then it will get filtered back into the circulation, and then the liver will also get a significant dose. And so while it's important to work at as low of doses and with the most specificity as possible in the promoters, obviously you'll still also need to screen for systemic toxicities, in particular liver toxicity, when you're doing a CNS specific approach. One final kind of side comment, because it comes up a lot, it's increasingly seen in primate studies, and people don't quite know exactly what to do with it yet, is the idea of dorsal root ganglion inflammation and toxicity. And so when you have use AAV vectors in the central nervous system on non-human primates, there has been the discovery of some significant pathological findings of inflammation in the dorsal root ganglion cells, along with some cellular degeneration. This is a very clear finding that is not controversial as a finding at all, but what is a little bit less clear is what to do with the finding, because while it has been seen in a number of studies, the animals that have this inflammation don't necessarily show clear symptoms clinically, and so it's the level of dysfunction that one could expect if this was also occurring in human studies is a little bit difficult to predict. As noted here, I mean, there's a number of things that might impact DRG pathology. It can be mitigated using an miRNA-mediated detargeting, and but at the moment, this is a finding that people are very focused on, that we're still kind of trying to figure out the relevance on, and I think a big component of that is going to depend on whether or not there are significant symptoms associated with it in studies going forward, or if it's observed in association with significant symptoms in any human trials in which the CSF is being dosed. And so overall, in terms of thinking about why gene therapy might be useful, I think that there are definite positive messages on the efficacy side, right? So there are 2 currently approved therapies that are demonstrating improvements in function, improvements in quality of life. When you look at the investigational gene therapies, you see examples of severe diseases in which patients are able to acquire function in a way that untreated patients essentially never do, and where there would be no expectation of acquiring that level of function in an untreated patient. The results in SMA and in some of the X-linked myotubular myopathy patients are very good examples of that. Similar experience with durable expression and rescue of phenotypes in hemophilia and in neurodegenerative disorders such as Sanfilippo. But at the same time, we need to understand that there are risks associated with this, and so there are significant possibilities for severe adverse events. It's seen across multiple diseases. It's difficult to model in animals, and so a lot of our learning is going to be in human patients. And really it speaks to the need for a balanced discussion of benefits versus risks, and for a maximal understanding of the disorders that are under study. As a person that works on the investigational and clinical trial side of gene therapy, I can say that this is, a very exciting therapeutic modality, a very exciting time to be working on this thing. And yet it's very concerning and very scary, because you don't want to endanger patients. You want to help the patients, and you don't want to endanger them unnecessarily. But I hope that at least these first 2 talks in this series provided some context for the types of work that we do, and some of our enthusiasm for what we think is a very bright future for this therapeutic modality as we move forward in learning how to make this more efficacious and safer for everyone, and move into other disease states and tissue types, including the central nervous system. So thank you very much for your time, and I hope you enjoy the remainder of the series.